so much. Thank you, all of you, for staying to the last day, and the organizer for managing us, and other people helping with the organizing this meeting. So I'm Ali Rahnavard. I'm from George Washington University. I'll be sharing with you an approach named Guide Tokenizer to enhance genomic language models to perform genomic language models on metagenomics data. So I've been recruiting audience for this cozy the past few days. Some of them are new to the microbiome, so I'm going to give them a little bit background, and then I'll go to the method that we are developing here. So as all we have seen in the ISMB, there are many omics data that we use to test different biological hypotheses, and many of these data are sequencing data, metagenomics or genomics, for example. And here I'm showing the number of studies that they have been published using these different omics data, using different technologies. And for the metagenomics data, I'm zoom into different omics that they are paired with the metagenomics and how the metagenomics data has been used for investigating the human health. And there are two technologies for sequencing data. We use the 16S and shotgun sequencing that you have heard of them here. And the key thing that in the microbiome field we are working on is in the genomic language models for the microbiome is to replace some of the tasks that we have been doing with the alignment. As all of us know, alignment has been a main task for many tools that we develop. Basically, you have a query, you map it against the reference you have, and you use that result to build these tools that we are showing. Some of the tools are faster, some of the tools are more comprehensive. And here we are trying to use the genomic language model to treat the DNA and sequencing data as a language. So my talk is going to be focusing on tokenizer. Here I'm showing that a general big picture of a genomic language model development. Usually we start with some source of genomics data, which I believe the genomics data compared to the language models, large language models, are more reliable because the data come from like well-known resources. We can extract DNA, we can tokenize them, and develop or model for different tasks. And here I'm going to focus on the tokenizer step that we can improve it and have a better result in the whole process. So my lab focus, the focus of my lab on genomic language models is to develop models that they are feasible, meaning that you can run them on your laptop or with less resources, and they are scalable, that you can run them for many sequences, and also they are accurate. And what we have contributed so far, that I'm not going to talk about all of them later, just I want to mention them, that if you want to take a picture, please do that, because there are a few preprints that I'm going to share. So one of them is to develop a roadmap, how we should tackle genomic language models where we are thinking about developing them, and how we can apply them to a specific domains. And then here at the end, I'm going to share with you the guided tokenizer as an innovation in our work. So one of my PhD students, Mahdi, he has done a great job really with a lot of effort he put into the genomic language models to see as a researcher what we should care when we develop a genomic language model. So I'm not going to go through the detail of all of this, but he has a preprint available, and this is a figure one from that preprint that share with us what are the important aspects of a genomic language model when you start to build them from scratch. So, and has a very interesting result that you can read the preprint here. And if you want to take a picture of this, feel free to do. If you don't want to take a picture, remember the SICK lens. So that's the name of the models he developed as a result, small models that you can ask a specific question of them. So also another PhD student in my group, Matthew, he used the result of the previous work and tried to apply them to a specific domain. So basically using the genomic language model to detect antibiotic-resistant genes. So there is a lot of challenge when you start to use the genomic language models for a specific task, not just only developing them. When you deal with a specific task, like the ARGs detection here, so there is many aspects that we need to take care of here. So the previous presentation, some of the tools like here we have in the right A, so for example, we have the alignment-based approaches and some machine learning tools, and we see the genomic language model for our case would perform those approaches. So, but the algorithm I want to share with you here is to how we can improve the tokenization, which is one of the deep steps and important steps in developing a genomic language models. So we have here in the previous talks that there are different tokenization approaches. One of them is the K-mer approach. Usually K-mer is 6. Here I use the example of 3 just to make a cartoon and share with those that they are familiar, they are new to the tokenization approach, how these approaches work. For example, here, the K-mers, you try to make all the tokens that in your sequence that they have a size of 3, and you see we have generated all the tokens with the token number, K-mer number 3. Then there is another approach which is more common, byte pair encoding, which tries to find the frequent patterns in the data and uses a merge. For example, GPT models, they use about 100K iterations to find these tokens. So what we are proposing in our approach is to keep the BPE and also find the tokens that they or purpose. For example, let's say you are trying to classify the reads that they come from different genus, and you know there is a specific token in each genus that is unique to that genus. So we want to keep those and force the algorithm to keep those tokens, and they might help us later with the classification step. And that's what we call it guided tokenizer. Basically, guided tokenizer is not a very specific task. Instead, it's an approach that based on your domain, you find what is unique about your domain, and you try to incorporate them in your tokenization technique, and then you can incorporate it in the model. So in the right, I'm showing a diagram how we think about, or how we have done this to generate these specific tokens, enforce the tokenization approach, and include them in our model to improve the performance of the model. So as you see here, for example, ATA, I just used a cartoon here, that is a unique token to one class. So I enforce my tokenization approach to not break down and keep it as a token, and then later I include it in my model and I give it a weight. There is a lot of detail behind what I'm saying here, but that's going to improve the classification if you are using a genomic language model for classifying the reads. So Vedant has done a great job. I told him if you produce the next figure that I'm going to show, I'm going to, instead of talking, I will be dancing. So, and he did it actually. So he made some changes in the last minute that showed what I want to share with you today. Basically here I'm sharing in the guided tokenizer, there are many ways to think about we try to use is to find the common tokens in each class. Then we try to share them with the weights we have in the model. And in all of the cases we had, which they had a meaning, they outperformed the BPE, and I'm showing some of those results that Vedant generated recently. And so I want to add one more comment before I show the next figure that requires a dance from me for Vedant, because he generated and it needs some like a last minute work. So the other aspect of our work and the guided tokenizer is to include the information we have about the domain. For example, in microbiome world, we know this tree of life, and we know each genus is under one family, and those families are under order. So what we can do, we can incorporate that approach in our fine-tuning approach. For example, we developed one model just to decide about the order, then we did a guided tokenizer for that order, then we move to the families, and we generate multiple models for each model, and then for each family we generate a model that decides about the genus. And this hierarchical approach actually improves our result as well. So my point here is that the knowledge in the domain, which here is the hierarchical structure between the species and or strains we have, so that helps also to improve the performance of the models. So I asked Vedant to use a community, a mock community data, and run our approach. We know what is the expected to see, which is in the left, and in the middle is what we performed, and in the right is the data too. So many of you are familiar with to do the profiling of 16S data. So 16S data, keep in mind that is an easy task for alignment-based approaches, because we believe that the sequences of the 16S are very ready. Okay, so here is the result. The performance is great. I see my time is close. I still have 1 minute. So the accuracy. So you see our accuracy increases compared to the alignment. Remember, I'm showing a case that is hard for our approach, which is using the sequences 16S against the genus sequences, which is easy task for alignment, still will outperform the alignment-based approaches. And the time. So these are the three things that we want to show here, the accuracy, or two here, the accuracy and the time, and both we are doing that. Okay, excellent. So I just want to share with you the summary of this talk. So I share with you how to generate a GLM language model, the domain, the guided tokenizer, and one specific application of that. The next, what we are doing is to look at the strategies to improve this approach and make it more detailed for each domain. So that's something we are working on it. So this work has been sponsored and funded by NSF. Now my university and NSF are funding it to commercialize some of aspects of this work with my co-founder, Keith Crandall. We have like a startup on this topic, 6side. And next year, the ISMB is in DC, so I invite you all to visit our lab, or if you come to Washington, so you can contact us and we can give you a tour about our lab and what we do. Thank you so much.
Thank you very much. We have time for a question. There.
Thank you for the very nice talk. Yeah, here. Okay. Yeah, my question is related to the token generation. Like, my question might be very naive. Like, how do you get the token related to one particular genome? You said it is very specific to strains, right?
Yes, absolutely.
So, do you have any kind of database where you assign each token to one particular strain?
Yes, I'll try to be brief because each of these steps has a lot of details behind them. For that, we do a comprehensive search, for example, for 16S to find the tokens that they are unique to those species. What is the benefit there is we can find the tokens that they are unique to each 16S using the database. But the good thing is after that, the model is database free and also learn from the databases because the end result uses the probability. Even if something is not in the database, we still can learn from the database and predict the closest label.
Okay, thank you. Time for more questions, but I'm sure he's available afterwards. We already have some delay. So thank you again.
Thank you.